Hello everyone, I'm Janine Gate Ramirez, a science major student of Mariano Marcos State University, College of Teacher Education, and today I'll be sharing the findings that we've gathered in the cell cycle. Cell cycle is an event involving cell growth and cell division to produce new daughter cells. The cell has two major parts, which is the interphase and the mitosis. During interphase, the cell undergoes normal growth process while preparing for cell division. The three stages of interphase are G1 or the gap1, S or the synthesis, G2 or the gap2. The cell cycle has checkpoints to ensure that the requirements of the next phase are fulfilled. During the G1, the cell increases in size and prepares to replicate its DNA. Toward the end of the G1, the cell has to be sufficiently healthy to replicate its DNA. Also, at the end of the G1 phase, a cell can exit the cell cycle when it receives a signal to differentiate or when resources are insufficient to grow and divide. The cell is said to be in G0, which is the resting or non-dividing stage. Through the S phase, DNA is continuously monitored for replication errors. If DNA synthesis progresses without errors, growth signals will stimulate the cell to proceed to G2 during the cell matures. In G2, the cell continues to grow and prepares for division. To proceed to the next phase, all chromosomes have to be fully replicated and contain no other types of damage. The last phase is the mitosis. In this phase of the cycle, the cell stops growing and divides into two daughter new cells, each with the same number of chromosomes. During prophase or the first phase, the nuclear envelope starts to dissociate into small vesicles, and the membranous organelles fragment and disperse toward the periphery of the cell. During metaphase or the change phase, all chromosomes are aligned in plane called the metaphase plate or the ectorial plane, midway between two poles of the cell. During anaphase or the upward phase, the cohesive proteins degrade and the sister chromatid, now called chromosomes, is pulled rapidly toward the centrosome to which its microtubule is attached. During telophase or the distance phase, the chromosome reach the opposite poles and begin to decondense, relaxing in the chromatin configuration. The mitotic spindle are the polymerized into tubulin monomers that will use to assemble cytoskeletal components of each daughter cell. Nuclear envelopes form around chromosomes and nucleosomes appear within the nuclear. Take note that if regulators or the checkpoints don't function properly, an organism may end up with too few or too many cells, and this can cause problems of varying severity from harmless hair loss or the growth of rats to the development of life-threatening tumors. So that's the end of the cell cycle. Thank you for watching and have a good day.